Hello everyone, and thank you for attending today's webinar, Gene Fusions in Cancer, Understanding the Basics of Oncogenic Fusion Detection Strategies. I'm Jade Parker, Senior Editor of Oncology Central, and I'll be your host for today's event. Before I introduce you to our speaker, I'd like to quickly run over a few housekeeping items. All the widgets are resizable and movable, so please feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right hand corner. Please let us know your thoughts on today's webinar by tweeting us at Oncology Central, or you can simply share today's webinar with friends and colleagues using the share widget on your screen. So some networks may cause slides to advance more slowly than others. Logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. So we are delighted to have Agnieszka Gajbros-Goniak, EMEA Technical Lead Oncology Staff Clinical Applications Scientist at Illumina, presenting for us today. Agnieszka is a Clinical Applications Scientist and joined Illumina in 2016. After completing her degree in biotechnology, she worked for various companies providing solutions for cancer research and diagnostics. Her current position involves specialist support of Illumina's oncology targeted and next-generation sequencing panels for detection of small variants as well as gene fusions. Thank you again for joining us today, everybody. So now I will hand over to Agnieszka, who will start today's presentation.
Thank you, Jade. I am happy to be here today. Thank you for joining us in discussing the importance of gene fusions in cancer and strategies for detection of these rearrangements. In today's webinar, I will briefly discuss mechanisms of fusion formation, listing some examples of well-known alterations. We will then talk about traditional methods of fusion detection, such as karyotyping and FISH, discussing benefits and limitations of those techniques. Finally, we will explore NGS-based approaches, and I will compare two methods of library preparation that can be used for detection of gene fusions, amplicon-based and enrichment-based. I will conclude with explanation how accurate detection of fusion events can help with the management of cancer patients. So let's start with the definition. Fusion is a hybrid gene formed from previously separate genes. Fusions may originate through balanced and unbalanced chromosome rearrangements. Balanced changes include translocations and inversions, and unbalanced intrachromosomal deletions. Translocation is the exchange of chromosomal material between two non-homologous chromosomes. A very well-known example of chromosomal translocation is BCR-ABL, which is present in around 90% of chronic myeloid leukemia patients. It occurs when chromosomal material between chromosome 9 and 22 is exchanged, and it leads to the formation of a small derivative chromosome 22, which is known as the Philadelphia chromosome. When the fusion gene is expressed, it forms a constitutively active tyrosine kinase, which phosphorylates multiple cellular targets and leads to uncontrolled proliferation of cells in CML. Interstitial deletion is a loss of chromosomal material in the interior of a chromosome. A good example of a fusion that is formed as a result of such deletion is TMPRSS-ERK. This gene fusion is present in around 50% of prostate cancer patients. It occurs when intrachromosomal deletion of 3 megabases on long arm of chromosome 21 brings together TMPRSS and ERK genes. Its functional role is not fully understood, but it is believed to mediate cell migration and invasion. Finally, there is a chromosomal inversion, and the very well-known example is ALK-EML4 rearrangement. ALK-EML4 is a gene fusion that is present in 4% to 5% of non-small cell lung cancer patients. Its prevalence is not as high as fusions discussed previously, but targeted treatment is available, so testing is important. It occurs as a result of 2 breakpoints on the short arm of chromosome 2. The fragment between the breakpoints is cut out and inserted in the opposite orientation, fusing the ALK and EML4 genes. Expression of the fusion gene leads to constitutive ALK signaling, which can contribute to increased cell growth, survival, and metastasis. Now that we have covered how the fusions are formed, let's discuss traditional methods of fusion detection. Karyotyping, also known as cytogenetics, is a method of routine chromosome analysis. With this technique, lymphocytes from peripheral blood or bone marrow cells are cultured using mitogens to stimulate the cell division. The cells are then arrested in metaphase using mitotic inhibitors such as colchicine. They are then fixed using fixatives such as Carnose and spread on a microscope slide. The chromosomes are stained using Giemsa stain, which produces banding patterns on chromosomes, and hence this technique is often called pre-banding. The chromosomes are examined microscopically, photographed, and rearranged into pairs for examination. You can see an example of such karyogram on the right. Karyotyping enables to observe abnormal gains and losses in chromosome structures. It allows for identification of various types of aberrations, including whole chromosomes, such as monosomies and trisomies, as well as large deletions, duplications, inversions, and translocations. On this slide, you can see 2 examples of abnormal karyotypes. The one on the left shows trisomy 21, which is linked to Down syndrome, and the one on the right shows BCR-ABL translocation between chromosomes 9 and 22, very common in chronic myeloid leukemia, also known as Philadelphia chromosome. The benefits of karyotyping include its ability to view the entire genome and visualize individual cells and individual chromosomes. This technique has been around for a very long time, and hence it's very well established and trusted. Also, it doesn't require any expensive equipment. The main limitations of cytogenetic analysis are its low resolution, typically down to around 5 megabases, but this depends on the quality of metaphases and sample source. For example, bone marrow samples from leukemia patients can be very difficult to analyze as the metaphases are very tight and banding patterns difficult to read. On the other hand, chromosomes from blood typically produce clear banding patterns, offering better than average resolution. Another issue is that karyotyping requires actively growing cells. It is therefore time-consuming, with the preparation of cells for examination taking several days. In addition, live lymphocytes are required, so blood samples need to arrive at the laboratory within a maximum of 48 hours after sampling, preferably sooner, to avoid failure of cell growth in culture. Also, the analysis is laborious and time-consuming. It usually takes more than 3 hours per sample, and it is typically done by 2 independent analysts. Next technique that I wanted to talk about is FISH, which stands for fluorescence in situ hybridization. And it is a molecular cytogenetics technique that was established in the 80s of the previous century. This technique uses fluorescently labeled DNA probes that hybridize to specific regions of chromosomes. They can be visualized with a fluorescence microscope in order to identify aberrations such as translocations, large deletions and insertions, as well as gene amplifications. One of the most important advantages of FISH is its ability to detect rearrangements in non-dividing interface cells. This enables short turnaround times with sample to result within hours. It is also compatible with many different tissue types, so not only peripheral blood and bone marrow, but also urine samples, sputum, and most importantly, FFPE tissue sections. On this slide, you can see some examples of FISH probes used in oncology applications. ALK-EML4 translocation dual fusion probe is used in non-small cell lung cancer patients to determine their eligibility for treatment with crizotinib or other ALK inhibitors. In acute promyelocytic leukemia, which is a form of AML, it is critical to test for the presence of PML-VERA translocation, as patients positive for this rearrangement require urgent treatment with all trans-retinoic acid in addition to chemotherapy to prevent brain hemorrhage that can be fatal. EWSR1 break-apart probe enables detection of translocations involving EWSR1 gene. Most common partner is FLY1 gene, but in small proportion of cases, EWSR1 can also be fused with ERK, ETV1, or other gene partners. And this probe is used in diagnostics of Ewing sarcoma, which is a primary bone cancer. FISH has a lot of advantages as it's been used for over 30 years. It is very well established and became a gold standard for detection of translocations. One of the most important benefits of FISH is its ability to detect rearrangements in non-dividing cells. So lengthy cell culture, as in case of karyotyping, is not required. It is possible to use interface cells and there is no need to have metaphases. It is also compatible with archived tissue such as FFPE material. It can visualize changes in situ on a single cell level. The workflow is very simple and quick, and it doesn't require any specialist equipment other than fluorescence microscope. Most probes available commercially call for overnight hybridization, so 16 hours, but there are also so-called fast FISH probes that can return results within a couple of hours from sample receipt. The biggest disadvantage is the analysis, which is time-consuming, subjective, and requires a lot of experience. Typically, 2 independent analysts review the signal patterns and then compare findings. The solution is much better compared to karyotyping, but it is still limited to around 100 kb. FISH only offers single gene testing, which can either be done sequentially or simultaneously, but in any case, there is very little room for multiplexing. FISH has no discovery power, only showing the status of genes directly targeted by probes. Next method is immunohistochemistry, or IHC, and it is widely used in the diagnosis of cancer. This technique uses antibodies to detect the presence of specific biomarkers or change expression of certain proteins. So these can be either enzymes, tumor-specific antigens, oncogenes, tumor suppressor genes, and tumor cell proliferation markers. IHC looks at the protein level rather than DNA or RNA level, but the presence or overexpression of a protein in a certain tissue can infer a genetic change on the DNA level. So, for example, HER2 overexpression in breast or stomach cancer is often linked to amplification of this gene, which can be confirmed with FISH on a DNA level. ALK presence in lung tissue is usually a result of ALK translocation that triggers expression of this gene that normally is only expressed in the nervous system. IHC is a technique that is very well established, it's cheap, relatively simple. However, it's not without limitation. The acquisition, handling, and fixation of the sample, as well as antigen retrieval and detection system, can affect staining. Moreover, the interpretation of staining can be quite subjective. Reverse transcriptase PCR, or RT-PCR, requires the knowledge of fusion partners and location of the breakpoint for assay design. Similarly to FISH, it has no discovery power, but due to high sensitivity, it can be used for monitoring of minimal residual disease following treatment. It is not a single gene test like FISH, but its multiplexing capability is limited. It can be performed as standard RT-PCR or quantitative PCR. So now that we have discussed the traditional methods, I would like to move to NGS-based approaches to fusion detection. Because most of the traditional methods I described previously have no or very limited discovery power and basically answer the question, is this particular fusion present in my sample, yes or no, the discovery of new fusions has been slow. With development of sequencing technologies, on the other hand, a large number of novel fusions has been described. The timelines shown in the slide very clearly demonstrate how gene fusions discovery coincides with the improvement of DNA sequencing technologies. The colored boxes in the top part of the timeline represent the year in which the particular gene fusion was discovered, and arrows below the timeline represent the year in which certain sequencing technology became available. Like with karyotyping or FISH, detection of fusions by NGS is primarily done on RNA level. The reason behind this is the fact that breakpoints typically occur within introns, and the breakpoint regions often span large stretches, so they can be difficult to pinpoint on the DNA level. In RNA, intronic sequences are removed during splicing, so sequencing only exons is sufficient to detect the breakpoints accurately. Detection of fusions on DNA level is possible, but it requires sequencing large stretches of intronic sequences, increasing the panel size very significantly, making it less cost-effective. There are two main approaches for detection of fusions by NGS, amplicon-based and enrichment-based, each of them having benefits and limitations. So it is important to understand the difference between them so that depending on the question that needs to be addressed, the best method is chosen. Amplicon-based approach is also known as method of opposing primers. RNA is used as an input, and in the first step of library preparation, it is reverse transcribed to double-stranded cDNA, which then serves as a template for target amplification. In the next step, fragments are phosphorylated and adapters containing sample-specific index are ligated. Then the libraries are amplified to boost the yield, which allows for accurate quantification. And after cleanup, the libraries are ready for normalization and sequencing. The biggest advantage of this method is its very short turnaround time. The libraries can be easily prepared within a day, with total time of about 6 hours and hands-on time of about 1 hour and a half. The limitation of amplicon-based method is that it only allows for detection of known gene fusion partners with known breakpoints. Enrichment-based method is also called hybrid capture. Here again, the RNA input is converted to double-stranded cDNA, and the adapters containing sample-specific index are ligated to double-stranded fragments of that cDNA. Whole transcriptome libraries are then amplified in the first PCR step and subsequently enriched for genes of interest in 2 rounds of hybridization and capture. At the end, there is one more PCR to boost the yield of final enriched libraries, and these are then ready for normalization, pooling, and sequencing. The advantage of this method is that it allows for detection of known and novel fusions. It is sufficient that the probes used in hybridization steps target just one of the 2 fusion partners, because fragments spanning the breakpoint will be captured during enrichment steps, and as the alignment is done to the entire genome, the analysis pipeline is then able to identify the fusion partner regardless of whether it is targeted by the panel or not, and irrespective of the breakpoint location. The main disadvantage is long protocol for library preparation, which takes about 2 to 3 days. So having reviewed the methods of fusion detection, both traditional ones and those based on NGS, it is worth asking a question whether or not NGS is the best approach for detection of all chromosomal rearrangements. Oftentimes, chromosomal translocations are responsible for the emergence of oncogenes encoding tyrosine kinases such as BCR-ABL in chronic myeloid leukemia, PML-VARA in promyelocytic leukemia, or ALK-EML4 in non-small cell lung cancer. As the chimeric gene is often expressed, these fusion events can be detected using RNA-seq-based methods. They can also be identified on a DNA level using FISH, for instance. An alternative scenario is where chromosomal rearrangements result in the exchange of promoter elements with subsequent transcriptional upregulation of the complete coding sequence of one of the genes. And some examples include rearrangements involving immunoglobulin loci such as IgH, IgK, and IgL, which dysregulate the expression of various transcription factors such as MYC, oncogenes such as BCL2, or proteins involved in regulation of cell cycles such as cyclins CCND1, CCND2, and CCND3, by bringing those genes under the control of globulin gene transcriptional regulatory elements. A very important thing to note is that these constructs are not expressed. Only the upregulated gene is expressed, and because there is no chimeric mRNA, Such rearrangements should be investigated on the DNA level using methods such as FISH or karyotyping rather than on the RNA level. So in this case, RNA-seq is not a viable option. Of course, it is possible to use DNA-seq, but as I have mentioned previously, that method is not really cost-efficient because it would require tiling entire gene sequences, including introns. So that would make the panel size really big. Let's now talk briefly about the importance of fusions in cancer. Despite the fact the fusions are not as common as single nucleotide variants in indels, chromosomal rearrangements resulting in fusions are present in approximately 20% of all human cancers. And currently, more than 1,000 gene fusions have been described in literature. Cancers that are primarily driven by fusions are hematological malignancies such as acute and chronic myeloid leukemia, lymphoma, and multiple myeloma, as well as soft tissue cancers such as Ewing sarcoma or alveolar rhabdomyosarcoma. But the fusions can also be driver mutations in some solid tumors, so for example, non-small cell lung cancer. An important contribution of gene fusions to the management of cancer patients is the development of drugs that specifically target the proteins encoded by those genes. The tyrosine kinase inhibitor imatinib, approved by FDA in 2001, was the first drug that was specifically designed to target a specific fusion protein, BCR-ABL, in chronic myeloid leukemia. The success story of imatinib has increased interest in developing additional compounds against the product of gene fusions, and several new drugs, all of which are kinase inhibitors, have been approved by the FDA during the past few years. An important example is larotrectinib, approved in the end of last year, which has been the first initial approval based on genetic mutation rather than a specific tumor type. So to summarize, many gene fusions are strong driver mutations in cancer and have provided fundamental insights into the disease mechanisms that are involved in tumorigenesis. The close association between the type of gene fusion and the tumor phenotype makes gene fusions ideal for diagnostic purposes, enabling the subclassification of otherwise seemingly identical disease entities. In addition, many gene fusions add important information for risk stratification, and increasing numbers of chimeric proteins encoded by the gene fusions serve as specific targets for treatment, resulting in dramatically improved patient outcomes. Gene fusions are also ideal for monitoring minimal residual disease following treatment. Before concluding the webinar, I would like to highlight a few commonly asked questions in the upcoming slides. Which input material can be used for NGS assays? Most assays can accommodate whole blood, bone marrow, fresh frozen tissue, or FFPE tissue. Why can't rearrangements involving IgH regulatory elements be detected with NGS? They can be detected with NGS on the DNA level using whole genome sequencing. Alternatively, a targeted DNA panel can be used if all intronic regions where breakpoints occur are tiled with probes. On the RNA level, they cannot be detected by NGS or otherwise, as the construct is not expressed. Only the partner gene that is under the control of IgH regulatory elements is expressed, often overexpressed. So by looking at expression levels of that partner gene, one can indirectly infer the presence of the rearrangement. What is the turnaround time for each of the methods discussed? Karyotyping usually takes 2 to 3 weeks as cell culture is required. FISH can be performed on uncultured blood cells or FFPE tissue, and typically hybridization time is around 16 hours and analysis in around 30 minutes to 1 hour per assay per sample. NGS can take between 2.5 days to 5 days, including analysis depending on which approach, amplicon or hybrid capture, is adopted. It has been mentioned that FISH has no discovery power and can only identify known rearrangements. What about break-apart probes where the fusion partner is not included in the assay? It's true that with break-apart probes, one can confirm the presence of a fusion that involves a driver gene. However, it's not possible to identify the fusion partner. In some instances, like in NTRK family, it doesn't make a big difference which fusion partner is involved, but in some cases, knowing the fusion partner is critical, as different fusion partners can determine different response to certain drugs. For example, in acute promyelocytic leukemia, patients with PML-RARA translocation can be treated with ATRA, while those with ZBTB16-RARA fusion have been reported as being resistant to ATRA. What data analysis solutions are available for NGS assays? Depending on the assay provider, these may be local or cloud-based. Usually a report with a list of all fusions identified is provided. There are also tertiary data analysis options where fusions are annotated and interpreted with intent to generate a clinical report. Thank you all for your attention.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value too. So I'd like to once again thank our wonderful presenter, Agnieszka, as well as you, our audience, for your time. So if you'd like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at Oncology Central. Thank you again for attending, and goodbye.